Something about the one.
Oh, wow. This is the room I come to remind myself my life has meaning. All my funding's up in 1 week, so it's like...
Oh, God, have mercy. I'm taking your cortisol level.
Exactly. No. And you know what? I'm taking Obesity.
Oh, they're probably messing up your cortisol level. Hello, good to see you again. Great to see you. Yep.
Yes, I'm really looking forward to these. So I'm going to sit right behind you, I think.
I'll sit right next to you.
I'd love that. I know you don't talk when the Orioles are in last place. George and my husband are both Orioles.
Okay.
And when they do well, it's worse, speaking of course.
Oh, okay.
No, and George came over once and they were winning. That's the primitive nature.
Oh, good.
So it's wonderful when they're losing because
Then I just said this one. Oh, I see.
no, he doesn't even follow the march.
Okay.
He actually
Okay.
So let's get started, so we have plenty of time. Thanks everyone for coming on what is an especially beautiful afternoon. We were even wondering about could we project the slides outside in the sun, but decided that probably wasn't technically possible. For those of you who joined us yesterday, you know that this is the second annual Gesell Lecture in honor of Arnold Gesell, the first director of the Child Study Center. For those of you who don't know Dr. Gesell, his work was very much innovative at the time of mapping out the ontogeny of behavioral development and how we really do understand how a 4-year-old is different from a 6-year-old, is different from a 9-year-old, and creating those methods of observation that allowed us to understand development across time. Development has, and our understanding of development has gone in a long way since 1911, when the Child Study Center was founded with Dr. Gesell and those early studies. And we now have a much more sophisticated sense of the biology of early child development and across the lifespan. But we often don't have the opportunity to come together as groups of scholars to think about child development and its interface with adult health, with all the things that are a part of this medical school. So hence this is why the Child Study Center, in cooperation with the Yale Center for Clinical Investigation, has put together this Gesell Lecture series. And we're delighted that Dr. Megan Gunner is our second Gesell lecturer. Dr. Steve Sumi in the back was our first. And maybe we can keep up this tradition of every year each Gesell lecturer comes back to join the next Gesell lecturer and keep these discussions going. We've been actually imposing an enormous amount on Megan and asking her to help us build our stress consortium, stress and adversity consortium, and at the same time give 3 talks and some workshops. So we're very grateful to her for doing that. Yes, I know. And it will be well worth it. But we really can think of no one better to be this lecturer. So let me just tell you just a tiny bit about Megan and then give the floor really to her. Her work actually exemplifies this kind of careful understanding and mapping out of development, but in this case, the development as impacted by early childhood adversity. She is a Regents Professor and Distinguished McKnight University Professor of Child Development at the Institute of Child Development in Minnesota, that Midwestern state there. And her work addresses the questions that are so central to our field of child mental health. That is, how early experiences, especially those in caring relationships, influence children's developmental trajectories and serve as a basis for lifelong health. As you'll hear, she's been focusing especially on populations of adopted children, looking again at these kinds of very carefully thought out developmental models. What you may not also hear is that Megan is an excellent communicator and has a passion for how you translate our science into effective policy, into effective communication with policymakers, because much of what we do really needs to impact the children and families that we most care about, and that are living in some of the most dire circumstances, the subjects of our research, but we also have a moral responsibility to do better by them. And Megan spans that interface in a very skilled and compassionate way. You may not also hear that she is an extraordinary mentor, and our field has really been seeded by the people that you have mentored that talk about you. And we, all of us actually count ourselves as grateful to you, Megan, and you're exemplifying that this week. So thank you so much for being the lecturer. Here we go for your second lecture, and welcome again to the Child Study Center.
Thank you. Well, I really appreciate you staying inside on this beautiful day, and I think you're all crazy, so you should get up and leave, right? And go out there. It's just gorgeous. So what I'll be talking about today, I know I see some familiar faces. So you were here yesterday, and you heard what I was talking about yesterday in terms of the behavioral sequelae, some of the key ones of children who have started out their lives in early deprivation and neglect. And today what I'm really focusing on is what I got into the field to study, which is how do we understand some of the mechanisms that help get those experiences under the skin and have, so that they have an influence on lifelong health. And I showed you this yesterday, if you were here. This has been produced by the National Scientific Council on the Developing Child. And yeah, Steve, I wish you'd gone out and bought that laser pointer for me already, because I'm having trouble with this. Okay. Not that. No,
Do you want
no.
me to get you one?
No, I don't. I'm going to point here. So the idea here is that early adversity through toxic stress, through environmental exposures, malnutrition, gets embedded biologically. That's what we're interested in. But how is it getting embedded? Right? We know that it's getting embedded in part because things are influencing the way the brain is developing, the neuroendocrine system, metabolic issues, cardiovascular, immune system. But in all of these, there's been a lot of focus on stress, this biology of stress, as part of what is helping to embed these experiences into the biology of many parts of the body and then influence over time your health and functioning. Stress, of course, is a multifaceted thing, right? And this is a beautiful paper if you're into stress. It's not that old. It was 2009, written by Marion Yoles and Tali Baram, called The Neurosymphony of Stress. Beautiful, beautiful paper. And they talk about all the different influences on stress and the multiple kinds of systems that you really need to be interested in. Now, most of these are operating in the brain. And as a developmental psychologist studying living children, I ain't got access to those. We have access to epinephrine through looking at the sympathetic adrenal medullary system in the periphery, and we have access to corticosteroids. So I have focused much of my career measuring those two things, but with a special focus on the hypothalamic pituitary adrenal cortical axis. And the reason I focus on the HPA axis is not only is it readily accessible, and I hate electrophysiology. It's just a nightmare. You got to hold the kids still. It's awful. But more importantly, I focus on the HPA axis and particularly on the protection from the adrenal of cortisol, because cortisol is such a powerful steroid hormone. It operates predominantly, though not solely, through gene transcription. The receptive elements for cortisol are on so many genes, multitude. Cortisol, being a steroid hormone, gets into every cell of the body. It has influences across numerous parts of the biology, and as a developmentalist, it participates in the development of the brain. It has big influences on changing the way the brain is functioning, because the genes that are involved in this regulation are in areas that influence memory, emotion regulation, autonomic reactivity, etc. In the periphery, I mean, if any of you are pediatricians out there, you should be worried and interested about this because it influences the lungs and the hearts and the immune system, the kidneys, and everything, which is, I mean, it's just a powerful hormone. And its regulation, therefore, needs to be highly regulated. It needs to respond when it needs to respond. It needs to shut off when it needs to shut off. And it looks like, from all of the research, 60 years or more of research on small animals, on the rat in particular, that the experiences early in life are critical for shaping the way this system activates, and therefore how it responds to all sorts of events that continue to happen throughout life. That, of course, was initially the research of a fellow by the name of Gig Levine, who was my postdoctoral mentor. A Gig, by the way, is a demerit in the Army, and his real name is Seymour, and he got Gig in the first day of being in basic training, and it really fit his personality well. But brilliant guy, and did beautiful research showing that if you handle an infant rat and you do it repeatedly over a certain number of days, you really have altered the way its HPA axis functions and its anxiety. That turned out to be a good thing, handling. Later, there was evidence that if you did it too much or too long and separated them too long, you could create the opposite of a neglected animal. And later, Michael Meany figured out that you could just take the high mothering rats and the low mothering rats, and you would see much of the same kind of thing, and it was an epigenetic mark on numerous parts of the body that was really altering and adapting that animal to the degree of support or harshness in its environment. Nature doesn't hate us. Part of this process is to help us survive, right? I started out with Levine. One of my earliest papers was a monkey paper looking at the effects of behavior and the HPA response during a prolonged separation in rhesus macaques. But I only did the work in Levine's lab because I wanted to come back and understand what was going on in humans. Sorry, there's Gig. And everything we knew, and actually at the time in Gig's lab, he was trying to figure out why I take the rats away and handle them for 3 to 5 minutes, and it has this huge effect over their life, and I'm doing it once a day for a few days. I mean, why is that having such a big effect? And they started talking about maternal mediation, because what they were doing was altering maternal behavior, and that lasted for more than 3 to 5 minutes a day. We know from the rodent work and the non-human primate work that early in development, essentially the regulation of the HPA axis involves the caregiver. You need to have the caregiver present and responsive to get a normal diurnal rhythm. That's been shown in monkeys. You take the mother away and the diurnal rhythm goes strange. You put the mother back and you get a pretty normal diurnal rhythm. We know that the presence and the availability of the mother or mothering figure is important for regulating stress hormone response, the HPA axis stress response. We know this in rodents, we know it in monkeys, we know it in humans, we know it in dogs and guinea pigs, and on and on and on. This is a very powerful regulator of stress responding, and it facilitates return to baseline after a stressor. So all of this thing requires having a responsive partner early in life. And if you come to my talk on Friday morning, you'll learn about the development of stress buffering in humans and what happens at puberty, and I'll just leave it there. Okay, a little shameless. After I spent 10 years figuring a lot of this out in humans, because nobody really knew how to study the HPA axis in humans. We had a few little studies. After 10 years, I thought, I know enough now that now I can start doing what I really want to do, which is understand how early adversity will begin to impact the development of the axis. And I had my first little foray into that with Dante Cicchetti. I sent a student to Mount Hope when he was still there. Jordan Hart, fabulous woman, a wonderful clinician in town in Minnesota these days. And we started studying salivary cortisol levels in maltreated children. And that was enough for me to say, oh gosh, that's just too complicated. Because these poor kids who are maltreated, bad things continue to happen in their life. And I was having trouble figuring out how we would sort out the impact of early experiences like my little rodents and the monkeys from ongoing adversity, which of course is the big question. We got to get there, but I wanted that first little piece. And it was about at that time, oh, sorry, there's my friend Dante in his gold lame jackets, which if you haven't met him. About that same time, I was invited to go to Romania, to Iași, Romania, with Tony Earls and Maya Carlson. And they wanted to collect salivary cortisol levels in the kids in the Romanian orphanages. And that was really interesting. Of course, we expected no regulator, really elevated cortisol levels. That's what we totally expected. That's what everybody expected. And we went and we collected the cortisol, and we saw for the first time what is going to be the theme of this talk, which is that the system does this bizarre flattening. So what we saw there was these are typically developing Romanian 2-year-olds. Who were in a daycare center, and the parents took the sample at home and then at noon and at bedtime, just to show you Romanian kids have a normal diurnal rhythm. 26 kids in this little red line, not one of them had a normal rhythm, not one. And while they weren't high, they were a little higher here and here, but it was this low morning, and generally we get this flat pattern. Now, if you're depressed, the clinical biological signature, HPA axis of depression, is high and flat. So that's not what these guys were showing us. They were showing us this low and flat thing, not one of them with a normal rhythm. I thought we'd messed up, but I got really, really interested. And Delia Vasquez, who I was running a network at the time, her nickname is Millie, so you might know her as Millie Vasquez. She studied kids who are deprivation dwarf. She's a neuroendocrinologist, pediatric endocrinologist, and she had data that looked like this with her kids who were neglected and maltreated in the home and were showing the stunting. So together we wrote this paper that said, hey guys, wait a minute, you might actually see low cortisol and a flattening of the expected rhythm in high-risk kids, and it may be an important risk for human development. And that was in 2001 that we published that. It's about the same time that Dirk Hellhammer started noticing that in adult studies, he would also see with chronic fatigue syndrome and other things, this flat, lower diurnal pattern. Okay. Now, time has moved on. And I want to convince you here that there now is a real accumulation of evidence that for many conditions of adversity, but especially perhaps for conditions of neglect, we're not sure that this lowering pattern is what you see. So this is work by Capitanio at UC Davis. They raise monkeys in a variety of different contexts, and they collect a ton of HPA axis stuff. This is after separation and relocation. This is in the afternoon. This is if you suppress the system with DEX, you're basically turning off the system at the pituitary level. And so you're doing that. This is stimulation of ACTH, so now you're jacking it at the pituitary level. And these guys, 1, 2, 3, all these 3 bars are different ways of living with your mother. Either in a field cage, which is a big enclosure, a smaller enclosure, corn crib, indoor individual cage with your mom. And these are the ones that are taken away from the mom and are raised in the nursery. So what you're seeing is these guys are not elevated. They're not elevated after you separate them for their cloth surrogate. They're not elevated in the afternoon. They suppress even more with DEX suppression. And they're not elevated if you go straight at the adrenal with ACTH. So overall, removing a baby monkey from its mother does not do what removing a baby rat from its mother, which is hyper. Man, we go hypo. Okay. And there have been recent, this I found just very recently. I said, thank you very much for doing this study. They did a meta-analysis of all sorts of forms of adversity, taking a look at whether you would see this kind of more hypo response. Now, if you're over on this side, it's more favors hyporeactivity, favors hyperreactivity. Many of these are null, right? Some are significant and hypo, but there's nothing up here. So in response to many of these studies, which have included adults and so on, as well as some kid stuff, but mostly adult. What you tend to see is either typical or low. Going hyper is hard, I think, for humans. And Andreas and I will argue about this later. But this is what these data looks like. So hypo is the thing that I'm going to be looking at. So I want to know, does the HPA axis stay hypoactive, functioning after conditions of deprivation are alleviated? Do you bounce back? Is there a sensitive period beyond which, if I remove them and put them in nice supportive homes, I don't see them bouncing back? Does it matter? Is it predictive of anything we care about? Probably is, but does it matter? And then I'm really interested in, and then sort of more current work, is there a period of possible reopening of the system so you can recalibrate it? Because the kids that I'm studying are these ones, this orphanage to adoption model, are kids who have grown up under conditions where they may get enough food and other things, but the amount of social care is really low, because it's really hard to care for a room full of babies and give them the attention you would give to your own child, or if you had just a few. And then, boom, they're adopted, and they're adopted into homes that are typically well-resourced. We've scored parent behavior. Yes, not all of them are perfect parents. There are a few that you would wish hadn't gotten the kid, but for the most part, they score very high on all the kinds of scoring we do for parenting quality. So you're removed out of that to a condition where you're getting enough food, you've got enough clothes, you've got enough attention. If you need help, you got the doctor. I mean, okay? Big, massive shift. How much recovery do we see, and how much repair of the system? Our first, there are many studies out there that are doing longitudinal work. I have actually decided to start, I've got a longitudinal study, you'll hear about it, but we started out by collecting and creating a registry of as many of the internationally adopted kids that we could get that was in the region close to the University of Minnesota. Minnesota was the highest per capita adoption state in the country. We have Children's Home Society. They, along with Holt, started in Korea ages ago. So we have a lot of internationally adopted kids. We did a survey. Initially had 4,000 on my registry. That's up to 7,000 or 8,000 now. They're aging out, though, and strangely enough, they don't want to sign up when they're 19 to continue. I think they'll start signing up after they have kids. They'll come, oh yeah, we'd like to know. They come from multiple countries because, although you've heard about the Romanian kids, that's the smallest place in terms of international adoption. China, Russia, India, Mexico, I mean, many. And I've done multiple different studies, so you'll be hearing a bit from different studies here. The first thing we did was actually a pilot study. I wanted to figure out I'd never worked with international adoption. I didn't know what I was doing. So we did a small pilot study where we looked at toddlers. They were 9 to 21 months of age. We matched them to children born and raised in Minnesota. This is, if you are a toddler, this is what your diurnal rhythm looks like. It's not fully developed because you still are napping in the afternoon. So you come down and then you're sort of flat on average across, and then you go down. And this is 2 months after adoption. So they look a lot better than the kids. If they looked like the kids, of course I don't know that. If they did, they're showing a much better rhythm, but it's still there. It's still lower significantly 2 months after adoption. Now, I told you that with Delia Vasquez, she studied kids who were stunted at adoption. Many are not stunted, who were deprivation dwarfs in their families of origin, neglect, maltreatment in this country. And part of the reason, she was an HPA axis person, and part of the reason is that there's a lot of talk between the growth system and the HPA axis system. Psychologists think that God put the HPA axis in so we could study it and know how people think and feel and so on. But really it's there to regulate metabolism and growth. That's its biggest function. And if you asked Mary Dahlman, she would definitely tell you that. So here's your growth system. You've got growth hormone releasing hormone that when it stimulates the pituitary, it increases the production of growth hormone. You have somatostatin, which is the brake. And when you stimulate that, it reduces growth hormone. Growth hormone interacts with cells in the liver to produce insulin like growth factor, which goes to your long bones and helps you grow, and to your muscles and helps you have nice muscles, okay? HPA axis, corticotropin releasing hormone, released in the hypothalamus, increases the production of somatostatin, puts on the brake. Cortisol interacts with cells in the liver to reduce its sensitivity to growth hormone. So what you have is a system under chronic stress. You've got a way to slow growth. Why would you ever want to do that? Growth is really expensive metabolically. And under harsh conditions, you may want to slow down. It's okay, be short and survive, right? Don't put all that energy into growth and then not have anything left to fight the pathogens and the other things that could be potentially be coming at you. And we certainly see that. Kids coming out of orphanages are typically at least one standard deviation below the mean, and many even reach criteria for a growth stunting, okay? So we got interested in whether, as an index of how harsh it was, and also as something that might reflect how much the HPA axis was activated and interacting. What would we see if we looked at the kids who were stunted at adoption? And this is a study that we published in 2011. The kids were 6 to 7 years of age at the time, and we had diurnal rhythm data on them. What you have here are the non-stunted kids and the stunted kids and the non-adopted kids. And as you can see, if you weren't stunted, mildly stunted, 1.5 standard deviations or more below the mean in height at adoption, your diurnal rhythm looks just fine. But the ones who came in stunted, and by the way, they're not stunted now. They're 6 to 7 years of age. You get that huge catch-up growth. They're within the normal growth range, but they're showing more. I thought I'd won the lottery. This is not always predictive. Sometimes the stunted ones aren't the ones. But it may be that what we were picking up here was the quality of care they were getting prior to adoption, along with genetic determinants that we need to pick up. Okay. Oh, and it also predicted inhibitory control. So if you were growth stunted and had this flatter rhythm, that was really a predictor of poor inhibitory control. Okay, so I've done a longitudinal study. We're in the midst of it. It's called Transition into the Family, or TIFS. The kids who were in this study were adopted later because I wanted to see issues between roughly 1.5 and 3 years of age. They came from orphanages, variety of places in the world. We saw them as soon as we could after adoption. We tried for 1 month. It was typically 1 to 2 months. About again at 8, 9 months, and then 16 and 24, because we wanted to map that period of transition recovery when you saw them going from having an IQ that's unmeasurable to having a normal IQ, et cetera, et cetera. We assessed them again at kindergarten, and then we had both a foster care, internationally adopted foster care kids in this group. They were adopted earlier because if you're a country that uses foster care, you get kids to permanent placement earlier than a country that uses orphanages, and a non-adopted comparison group, so with the same age. What we did in terms of the HPA axis was at each of these 4 time points, parents did 3 days, they hated it, but they did it, of salivary cortisol at wake up, at noon, and at bedtime. Okay, so I had 3 days of sort of diurnal pattern each time. We expected that we would see a gradual recovery. We did not. What we saw at the first time was what we saw at the second and third and fourth. And it's not going to look like much. But in fact, both my foster care and my PI kids, post-institutionalized kids, were showing a reduced morning level, just a little bit. Those are actually error bars hiding behind there because we had 12 measurements of the cortisol levels. But they are slightly reduced. What is predicting it? I could look at that for my post-institutionalized kids. And this is above and below, 1 standard deviation above and below the mean for quality of care prior to adoption. And this was the parents describing what they saw and a social worker rating it in terms of quality of care. And this is social care, not physical care. So you can see that my kids who were getting lower poor quality social care, more neglect, were actually showing a much more blunted, flatter pattern. And the other guys are looking pretty much like my non-adopted kids. Okay. Now, a flatter diurnal rhythm, we sometimes even just a little, we end up saying hypocortisolism. But that's not what really hypocortisolism is. Oh, sorry, forgot this one. When we were getting the initial data, we decided to figure out, was it just kids coming from orphanages? And I was running this lovely network at the time, and Phil Fisher, who studies kids in the foster care system, was part of the network, and so he went back, and he's got cortisol in his kids. And these are children coming into a foster placement. They're 3 to 6 years of age. Many of them had been in foster care before, but they're 3 to 6 years of age. And we looked at their diurnal rhythm. About most of them, 62%, had what looked like a pretty normal rhythm. They just didn't wake up in bedtime. 4% looked elevated, but 34% were below a standard deviation of my typically developing kids' mean. So they were flat. So they're seeing the same thing. And when they went back and did the Manley and Cicchetti rating of what kinds of experiences these kids had, it was severity of neglect that predicted it. It was not physical abuse or sexual abuse in these young kids. It was severity of neglect. Okay. So now on to the fact that you want to talk about this as hypocortisolism, but in fact, hypocortisolism should have to do with reactivity of the system. So what do we see if we start asking about reactivity? In that study, my TIFF study, where I've got all that cortisol data, I did also bring them into the lab 4 times, and we did everything under the sun with them, including trying to scare them, trying to see how they handled highly arousing stimulation. We slapped electrodes on them and measured the PEP and vagal tone. And the 4th time we were there, we said, no, we're not doing that. We slapped electrodes on their heads and looked at EEG asymmetry. Did a lot with these poor little guys. And they reacted to it. So the first thing to look at are the blue line, because these are my non-adopted kids coming from homes of well-educated parents with high incomes, like the families that adopt internationally. And they put up with what we were doing for a while, and then this is about where we did the scary toys, and then we slapped the electrodes on, and they responded. This is time 1, first time they come in. But look at my foster care and my post-institutionalized kids. They're a little higher to begin with, significantly, and then they just stay basically flat. Okay? That's hypocortisolism. And if I look across time, it gets really interesting. So as you get familiar with this, every time we tested them, we did roughly the same pattern of things, right? And what you're seeing for my non-adopted kids, here's that first time, and second time, and third time, and 4th time. They're just perfectly. Now, yes, they're getting older, but they ranged from 1.5 to 3 years of age at this first time, and that wasn't, so it's really experience. They're adapting. If you look over here at the kids from foster care, they're doing the same thing, even though they're a little flatter. Is that me?
No.
Oh, that's okay, good. Never mind. I did once give a talk. I gave a very big talk. They'd given me a Regents professorship. It was graduation. Thousands of parents in the audience. I'm giving a talk, and my phone goes off. And I'm going, whose phone is it? It was mine. And it was my kid, of course. And I got the biggest laugh I've ever gotten. I said, it's okay. I could call him back, but the answer is it's in your room. Every parent knew what I was talking about. But look at these guys. So this is their first time, second time, third time, 4th time. It's not significantly different. They're not really changing. But if anything, if they're beginning to, anyway. So we did a factor analysis. There are 2 factors. And the first factor is like 88% of the variance, the wake-up values at home, the slope of the diurnal rhythm, and the slope of the response in the lab. This is a hypocortisolism factor, okay? The lab arrival, I didn't expect it to go on there, and it didn't. So now we've got to measure what I feel pretty confident of is the degree to which they're showing this hypocortisolism response. And this is what, for those of you who heard before, this is what we used in these kids when we had kindergarten data to see what it would predict. And what it's predicting, hypocortisolism, controlling for sex, is predicting teacher reports of externalizing, more hypo, more externalizing, more problems with attention regulation. And other people are seeing this pattern as well. So, okay, it goes hypo. Is it just me? One way to look at, and really this, what I was doing with the little guys in the laboratory was not a really classic stress study. I was just wearing them out, really more than anything else. So you want to do a real stress study. And the thing that we know that is ethical, that you can do in the laboratory, that produces really lovely responses, is to go through a self-evaluative stressor, and the best one we know of is giving a public speak talk, right? So the Trier social stress test, they come in, they're told they're going to give a talk, they have 5 minutes to prepare it, then they get up and they give their talk for 5 minutes, and then after that they do mental arithmetic, designed to hit a level where they're going to make mistakes. The judges are supposed to, with kids, they're not supposed to be nasty with adults, they get a little nasty. But with kids, they're a teacher and they're being serious and they're taking notes, kind of thing. And we're not the only ones to have looked at this with kids coming from orphanages. There's a project in Romania, the Bucharest Early Intervention Project, where they did baseline assessments of kids in orphanages. They randomly assigned them to foster care intervention that was supported by Charlie Zena, beautiful intervention, or care as usual, which was to stay in the orphanage until Romania developed their own foster care system. And then they had a comparison group of never institutionalized kids who were born and raised in Bucharest, okay? And when they did this Trier social stress test, here you've got baseline, they did have a peer interaction, they did this Trier social stress test, they had a little frustration thing and recovery. This is what their never institutionalized Bucharest born and raised kids did, which is exactly what they were supposed to do from the standpoint of the HPA axis. These are the kids who were care as usual. These are the ones that were in foster care. So they get this hypo. They found hypo too. Okay. So it's not just us. Hypo-responsivity seems to be what this kind of at least serious neglect, which, by the way, I want to remind you, is exactly what they were doing to the rats and the monkeys that were going, and the rats were going hyper. Monkeys weren't. Right. Okay. Is there a sensitive period? Right? That is the big question. Where you're going to, if the kids are sort of provided with appropriate stimulation support, you wouldn't see this. And if not, you begin to see it. So that's the $1,000, $1 million, $1 billion question. In the Bucharest stuff, their median age of randomly assigning them to foster care was 2 years. Okay? In this study, if they looked at the kids before and after 2 years of age, so these are just the foster care kids, what you're seeing is that the ones who came out before 2 years of age, roughly a year to 2 years, they look pretty normal. They're showing a response. These are the ones that were 2 and over, basically 2 to 4 years of age at the time they came out. These kids, by the way, are 8 to 10 at the time of the assessment. Okay? So they say 2 years of age. Okay? All of us are using median splits because you need to have enough kids. And so the time's going to keep moving around here. But not that much. So one of the things that we've been doing is this question about social buffering. We stole a page from Clemens Kirschbaum, who'd been studying this in adults. We used the Trier social stress test. In that 5 minutes when you were prepping for your talk, we had the kids prepare with their mothers, or with their primary caregiver, most of the time the mother, or prepare with a researcher. So we just compared those two conditions. And what we found for children, pre-pubertal kids, is that preparing with your mother completely knocks out the stress response. So your mother's not watching you. As soon as you're done preparing, she goes to another room, you go in and give your speech, all the rest of that. But prepping, this is the period of time where you're getting the axis jacked up, it turns out. And your peak response actually needs to be timed from from the preparation period, not from when you stand up to give your talk. So we knocked out the stress response for prepubertal kids by giving them their social buffer during that time. What happened to the kids adopted from orphanages? Well, this is with mother support, and these are the prepubertal kids. There's my non-adopted kids. There's my post-institutionalized kids. This is preparing with a stranger, and there's my, excuse me, let's get this correct. These are actually, sorry, this is wrong. These are my non-adopted kids, and these are my post-institutionalized kids. No difference. If anything, and we couldn't get it to come out, they actually look like they're upset by being separated from the mom, but we couldn't get that to come out. So no, we're not seeing an effect. But remember, this is the sensitive period portion of my talk. The median age for the kids in the post-institutionalized group was 18 months, so we split at the median. And split at the median, these are the kids under 18 months. Everybody's here with your mom, okay? So, or excuse me, this is parent with your parent or with a stranger. So under 18 months, you're looking like my non-adopted kids. With a stranger, you're showing a reaction. Over 18 months, there's no difference between these two lines. So, okay, 24 months in Bucharest, because that was their median. 18 months, I'm seeing something's going on here, maybe. We've also been looking at this thing called the cortisol awakening response. So the way nature has figured it out is waking up is a challenge. No. So you show this diurnal rhythm where you actually have the highest level of cortisol roughly 30 minutes after awakening, and then it decreases with a little blip around the noontime meal because that's part of regulating insulin. And then you go all the way down, and it keeps going down over nighttime sleep, and then it starts to go up before you awake and REM in the last part of your sleep, and then it does that jacking thing after you wake up. Okay. And that time from 0 to 30 minutes, 34 minutes, it will increase about 40%. It seems to be part of what helps you wake up and say, oh, I think I'll go hunting for carbohydrates. And so, I mean, it's serving a function, again, more food, right? So we were but it seems to also, it's not part of the diurnal rhythm, it's actually part of something else, and it may have to do with sensitivity of the adrenal to ACTH, possibly. We're not sure. Okay, cortisol awakening response. We had this is a study I'm doing now. 139 comparison kids, 61 children adopted before 16 months, and 59 from 16 months to 5 years. Okay, so our median, obviously, is 16 months in this study. Kids are average of 11 years of age. They range from 7 to 14 years of age. And we had the CAR, cortisol awakening response, on 3 days, is what we're looking at. And what we found, this is the average of 3 days, but we did it with fancier stats. Same story. This is just easier to see. What we saw is these are the CAR for my non-adopted kids, for my adopted 16 months and under, or under 16 months, and my adopted 16 months. Okay. So I've got a blended CAR. So we've got multiple things going on, but again, it looks like there may be this sensitive period for being able to recover that is sometime in the second half of the second year of life, maybe. Okay. Now, however, it may also have to do with how bad it was. So we have data on children who were adopted very shortly after Ceausescu was shot in Romania. Those kids, the conditions during the last part of Ceausescu's era, in fact, the reason that it ended up getting shot is the country was absolutely in a mess economically, right? Which meant that children got the worst of it, and children who didn't have parents to protect them got even the worst of the worst. So the conditions in the orphanage were horrific at that time. Many of those children were adopted into the United Kingdom. Mike Rudder was asked to study them. He did. They're continuing to study them. They're young. They're about 25 to 30 years of age now. And at this last assessment, they looked at the cortisol awakening response. Okay? Now, generally speaking, in that English and Romanian study, kids who got out of the orphanage before 6 months or before looked pretty good. Their behavior was not particularly challenged. Those over that, yes. But this is different. Oh, sorry, why am I set myself up for all of this and then I screwed up? Wait a minute, where is it? Oh, it's up there. I'm going to jump to it since I set it up. So, English and Romanian study. What they saw there, and this is two ways of looking at it, they had wake up 30 and 45 minutes. These are the United Kingdom kids. There, they had a nice car. These are adopted under 6 months and adopted over 6 months. So both of those groups. So if there is a sensitive period for affecting the car and affecting it long term, maybe here it got affected really early because it was so bad. I don't know. But they weren't seeing any kind of recovery in the car. I've been interested in whether puberty does allow you to have this possibility of a recovery period. We certainly see marked changes in neuroendocrine activity with puberty, and not just sex hormones, right? We also see it with the HPA axis. It increases in its activity, or at least in its level, in humans. In animal models, there's beautiful work by a fellow named Romeo that raises the possibility that the peripubertal period is another sensitive period for setting the axis. Of course, in the animal work, what they're doing is doing bad stuff to the animal and finding out that if they do it then versus doing it post-pubertal or later on, early adulthood, the pubertal ones carry it forward and the adult ones don't. So they're looking at bad stuff. But I'm sitting here thinking, well, plasticity cuts both ways. So if it cuts negative, it might also cut positive. And perhaps if you're a kid coming out of an orphanage and you maintain the activity of your axis like you were living in harsh conditions, perhaps puberty will open up a window that would allow your body to sample the diversity around and reset that system. Which would make sense because it's sort of right before reproductive period, right? So maybe that's the pubertal recalibration hypothesis. The first glimmer we got that it might work, and that got me thinking about this, is in a study where we also were looking at the cortisol awakening response. And here we took a Rondall model. 12 and 13-year-olds are great for studying puberty and trying to unhook it from age, because you've got some 12-year-olds that look like they're 20, and you have some 13-year-olds that look like they're 5. Right? It's this period of time when some have really started and gone a long way. And so what Ron's model is you call them on the phone, you ask a few questions about puberty, you take your 12 and your 13-year-olds and you try to balance them for where they are in puberty. So you get rid of age as a confounder for this. So is it age or is it puberty? In here, it would be pubertal stage. We looked at wake up in 30, we had 60. This is not part of the car. So what you're really looking at here is the car part. And if we look at the kids who are pre-early puberty, these are non-adopted kids, they've got a car. These are my adopted kids, they don't got a car. Okay. We look at 12 and 13-year-olds who are a little further along in puberty, and we've got a car in both groups. It's a cross-sectional study. That's why I'm doing the longitudinal study now. And that's my qualifier that maybe it's not too terrible. So I'm doing this pubertal recalibration study now. It's the same one I showed you the car results for before. 139 kids, 7 to 14 years of age, nurse exam for Tanner, car on 3 days, which I showed you from the first year, cortisol to the Trier social stress test. We've analyzed the first year of data, okay? They're moving on. Before I show you the results, many of you have probably learned or heard that early adversity will make puberty hit earlier for girls, right? Which I would need to take into account if it was true. But I've got the best pubertal data because I got nurse's exam. I don't just have parent report. And what we see, is no effect on puberty. So in this first year, and we've analyzed the second year and it's still true. So no effect on pubertal timing. So I don't have to worry about that. But also the poor student who analyzed it, this was going to be her claim to fame, and she was really depressed. But it got published right away because it's the best study of this. And I said, it's okay. The people out there in that world will not hate you, I don't think. Okay. That model also predicts we're going to see obesity as a function of early adversity. So no, the answer is, again, poor student. Their kids are a little bit shorter They're leaner. We put them in the bod pod so we could look at lean to fat body mass. They actually have greater lean to fat body mass. They have lower BMI. It's at the low end of normal. There's no difference in waist-hip ratio. And it was also true of the ones that were stunted at adoption. So I think this is a hopeful, move them out of that direct into supportive homes, and maybe we've gotten off that trajectory. Maybe it takes the whole schmear, okay? But I don't have to worry about obesity in the work that I'm doing. So this is the Trier data from that first year. No, we didn't see it as CAR, okay? But maybe that will come a little later. These are the kids, again, controlling for age and stuff. These are trying to control for age. These are the kids who are pre-early puberty and also a little bit younger. And what you're seeing, these are my PI kids. They're showing no reaction. These are, you're supposed to adapt. You need to adapt and then respond. They're looking fine. Those are my typically developing kids. These are the ones who are older. No significant difference. So maybe, at least with regard to the stress response, we really are seeing a possible recalibration. But I won't know until I have a chance to see the same kids as they go through, and these are cross-sectional. I want to see the longitudinal. Okay, so good. I did it again with 10 minutes to chat. Childhood adversity, and perhaps especially, but we don't know for sure, deprivation and neglect, is associated with a low and more blunted activity in primates, including humans. There might be a sensitive period for setting this up, which if you can get out of this adversity and into a really good place sometime in the first year and a half, maybe, but it may have to do also with how bad it was, right? So there may be both things. Hypofunctioning of the HPA axis following early deprivation and neglect seems to predict problems with attention, regulation, externalizing behaviors, and probably in general with the development of prefrontal cortex. Because we also see issues with, you weren't here, but with all sorts of executive functions. And maybe we've got an opportunity to recalibrate the axis around puberty. If we do, it's probably going to interact with the conditions around puberty. And some of these kids are experiencing incredible amounts of psychosocial stress because they can't make it work with their families, their friends, or at school. And some are not. So we're also doing extensive stress interviews to see if that modifies it. And many people helped me get all this kind of work done. So thank you very much, and we have time for questions.
Any questions? George.
Yeah, I'm wondering if with respect to hypocortisolemia and also the CARs.
Yes.
Did you look at that with respect to sleep quality or time?
Yeah, we do have that sleep data, and it is controlling for sleep. It is not the thing that's doing it, though it might be part of what is getting it established. But no, it's not sleep quality. It could be sleep quality, but it's not sleep duration.
Good question. Jim.
So I guess we've been fascinated by hair cortisol.
Well, that's cute.
And I guess I'm just curious in terms of the hypocortisolemia, if you've also been looking at all at sort of the diary we have of our stress by looking at the hair cortisol.
We're beginning to collect hair cortisol now. I'm always slow on the uptake. I want other people to make the expensive mistakes before I do. So we are collecting hair cortisol now. We're doing it in a study of prenatal stress. We're working with Clemens Kirschbaum. No one's ever checked the reliability of hair cortisol. We know if you take it from different parts of the head, that's not good, but what's the reliability in the strands that you're actually assessing? And does your reliability get reduced the further out you are because of hair washing and so forth? So we're looking at that, and we're also looking at trying to measure it month by month in hair cortisol and hair. I wished like the dickens that I had known about hair cortisol when these kids first arrived. If you're interested in doing research on internationally adopted kids, don't. The U.S. is down now to less than 5,000 kids being adopted a year from a high of 25,000. There are just no kids coming in. for a variety of reasons. Some of them very good, like places are developing their own system. They're changing their own population's belief about adoption. So they're trying to encourage, like Korea, yes, it's okay to adopt someone you're not biologically related to. A family, change your meaning of family so these kids can get adopted within country. Some for reasons that are really horrible, because there were new rules on what families or what countries had to do in order to be able to be certified to have adoption happen, and everybody thought that that would slow it down for a few years, and it just turned the spigot off. So there's just nobody coming in. But I wish I had, because that's when, if I could have looked at their hair when they arrived, right, that would have been the thing, then to be able to really see, or so what's happening before they got here. But hair is really an interesting possibility, especially for conditions where you aren't able to be there at the time of the challenge that you are interested in.
I guess the other issue too is about the gut-brain axis and our microbiome, and I'm sure you've been reading about that too.
Oh, yeah. I've got a lot of poop in my lab right now. Actually, Jane Foster has it, so I'm collaborating with Jane Foster. We've been looking at the immune system in kids that are teens, because they'll let us bleed them, right? And the IRB will let me ask them if I can bleed them, and they're like, yeah, go anyway, give me money. And so we've been looking at teenagers adopted from orphanages or not. We've been doing complex immunophenotyping, as well as stimulating the blood to look at response to various like LPS and so on. And we've been collecting poop and sending it to a microbiome person in Canada who's willing to do it for free. And we'll be seeing soon what we're seeing in the microbiome, if anything of interest. Yeah. Ooh, Amy.
As long as you're talking about blood, do you have any catecholamine or cytokine measures?
Do I have catecholamine or cytokine measures? No, not those, but I do have PEP. I have pre-ejection period, which should be reflecting epinephrine. Are you guys familiar with pre-ejection period? Okay, so basically, if you send a little electrical signal across the heart, if your heart is full of blood, the signal is faster than if your heart isn't full of blood, and your heart fills and squishes, basically. You've got the sinoatrial node, right, that tells your heart when to beat, and then there's another valve that actually opens up. And that time between the beat and whoosh is what you're trying to get at, and that is regulated by epinephrine, not norepinephrine, epinephrine, primarily. And it's adrenal rather than other nerve ending. So it's a much better measure of the sympathetic adrenal medullary system. So it's one catecholamine. And we've looked at that and in these kids, and it's a little different, but it's not a lot different. It's not a lot different. I'm not seeing it in the cardiovascular system or in the other measures of autonomic function. The big signal that I'm seeing in the HPA axis. And if anything, you've got more sympathetic, which is what you would expect. That's what you're expecting. What I was expecting, a little elevated, sympathetic, hypo HPA axis, right? That's more the pulling the whole system apart.
So
Yeah.
sort of related question, whether in your research or in others, looking at groups that have a flattened cortisol rhythm, have people also looked and seen flattened rhythms, say in melatonin or in epinephrine or in norepinephrine?
Yeah, that's the question of how, is it more than the HPA axis?
Yeah.
There is not much that I know on that. If I were to go after a low-hanging fruit, I might do heart rate at different times of day or body temperature or other kinds of diurnal rhythms. It's not necessarily, that's why somebody asked me about sleep. It's these rhythms. If I were able to be in the brain, I would expect that actually the HPA axis is following a pretty normal diurnal rhythm, and it's downregulated, so it's producing less power. There's a downregulation of the core production and not of the diurnal. So, yeah.
Thank you for a wonderful talk. I have a question whether the measurement or the phenomenon of flattening
Yes.
that you described, does this happen primarily when you take the children out of the adverse environments and put them in a more optimal environments, or whether you have
We saw it
measured-
in the orphanage.
They are still in the orphanages.
We saw it when they were still in the orphanage, even flatter, and it actually got a little better once they, well, excuse me. Kids in the orphanages look flatter than the kids we've looked at who are already in their homes. Wish it was the same kids, but, yeah. One more question?
So is CAR as, I mean, I guess we could debate how robust, but at least diurnal flattening has been semi